So good morning, everyone. For those of you who have not yet met, my name is David Brown. I'm president of MGH, and I'm really thrilled to be here at the second meeting of our Cancer Center Advisory Council. We held our first back in May. I didn't realize actually at the time that that was your first. I just knew it was my first. So it turns out I've been at all of these. So I'm in now my second year as MGH president. And as I've said to each of you, almost every person in this room that I've already met, cancer really is my top priority. And I see it as our collective responsibility, even in times of headwinds in healthcare, to double down on our commitment to cancer, the science of cancer, the clinical care of cancer, the education of the next generation of cancer scientists and cancer physicians and caregivers, because it's really cancer that impacts our society the most, and we are really at the precipice of groundbreaking work that will change the way patients experience cancer, and you'll hear some of that today. And I'm really sorry that I'm not able to stay for the bulk of this presentation because I really want to hear about this work, and I've already told Daniel Haber that I will be coming back to visit the labs and to learn more about this incredible work. So I do want to take a moment to thank all of you for joining us and for your commitment to cancer. I travel with this little model. I'm pretty sure I brought it to the last meeting. So those of you who are here at the last meeting remember me. I travel around with this all the time, and I'll talk about why in just a moment. So as I mentioned, a little later in the event, you'll hear about the future of CAR T-cell therapy, including a real robust group of experts and one of our patients. So I want to thank Marcella Moss for leading this effort and for sharing her expertise with all of you in just a few minutes. And then we'll hear from Dr. Matthew Freegalt, who's the clinical director of the cell therapy program, and we'll hear from one of his patients, Jill Dachko. So appreciate all of you being here today to create a special experience for our advisory council, and I think it'll be informative and inspiring. So as I said, cancer really is my top priority, and it's certainly among leads the way in terms of our clinical commitments. And some of that is based on what I just told you, that the science is about to take us to new places, and we just have a commitment at Mass General to push that science. I think more than a commitment, a responsibility, because if we don't step into that, which academic medical center should? I think that it's also important to recognize that every day at Mass General, we turn down requests for transfers of about 30 patients in our region, patients who are in other hospitals who can't be managed in those other hospitals. And I wouldn't say the majority of those patients, but the plurality of those patients have cancer. And so we don't have capacity right now to meet the needs of our community. And part of why I'm excited about this and our new building is that we are going to rebuild and create a true integrated cancer center on our campus. I think one of the special things about our cancer center is that it is embedded within a general hospital. And again, just as I was saying at the table I was sitting at, patients don't experience cancer as an inpatient or as an outpatient. They live with cancer, and sometimes they're sick enough to be in the hospital, and sometimes most of the time they're well enough to be home, and sometimes they're somewhat in between. And this new building is going to create the structure for all of that experience: outpatient care, short stay care, and inpatient care all in one center. And so that's an enormous opportunity for us. I just can't say enough about the leadership of our cancer center and the opportunity, I think, to really lean in during this next generation where the discoveries will change the way our patients experience cancer. So because we already can't take care of our community, we need your help. We need your help advising us, guiding us, and helping us raise the funds so we can put up this building. So when we last met, I think I had this model with me, and I was telling you how I was eager that later that month in May, we were going to get approval for the new building. We did, in fact, get that approval. And if you've been by the campus on Cambridge Street, you see that there's a construction fence up, and a whole couple of old buildings have been taken down, and we're starting to dig a hole in the ground. And by the end of this calendar year, there'll be a slurry wall poured, and soon this structure will grow out of the ground. First, so if you're looking at this is Cambridge Street here. This is Blossom Street and the hotel. For those of you who are MGHers, Harvard Gardens is across the street right here. Easy access for anyone working in the new building. This is going to be the most important building that we undertake at MGH, perhaps since the original building was built in 1811. In addition to, of course, building this enormous cancer center tower to allow us to take care of patients, it'll house the Corrigan-Minnehaha Cardiovascular Center, and it will allow us to decant where our cancer patients are currently taken care of in the Lunder building and expand our neuroscience offerings. So our three big areas of investment will be in cancer, cardiovascular, and neuroscience during the next decade. So many of you have been fundamental in ensuring that our science is on the leading edge, that our clinical care is on the leading edge, and you've been advising us in this forum and in others. I'm grateful for that. I look forward to many more of these meetings. I intend to be at all of them. I didn't realize that was a possibility, as I said earlier, but turns out it is. So before I hand it off to Daniel, maybe just a couple of general hospital updates, since it's now been 14 months since I've been president, and kind of looking back on the year. It's been a pretty challenging year in healthcare overall. COVID is on the wane, but the impact of COVID and virtually 3 years now of COVID on our staff has been enormous. Our staff are tired, and some of them are thinking a little differently about how they want their work to be part of their lives, and I certainly understand that. We came out of a really challenging fiscal year in 22. We hope that FY23 is a little stronger as we rebuild our workforce and get back to the business of taking care of patients across the spectrum of their healthcare needs, not just focused on COVID. We had a terrific year in development. I want to acknowledge the development team that's here, helping us raise money for critical programs that otherwise simply can't move forward. And some of the work that this group has supported in cancer certainly falls into that. It's also been a great year of building and rebuilding my own leadership team. We welcomed a new chief medical officer in June, Dr. Will Curry, who's a neurosurgeon, actually takes care of patients with brain tumors. And he jumped right in June and has fully embraced the role of chief medical officer for the hospital and for the physicians organization. And earlier, late last month, we announced the new department chair for pediatrics, and she'll be joining us on January 1st. I was really happy to recruit Dr. Joanne Wolf, who actually is a pediatric hem-onc specialist, cancer specialist, and with a focus on palliative care, which I'm sure will resonate with this group. She comes to us from the Dana-Farber and Children's Hospital. Of course, she's a professor at Harvard Medical School, and I suspect she'll get to know some of you in this room, given her interest in and expertise in pediatric cancer. And we're thrilled to recruit her. And I would say that, frankly, I'm thrilled to recruit a woman department chair. One of the things that's pretty obvious to me at my department chair meetings is that we are not diverse in terms of gender, and that needs to change, and it will change under my tenure. So happy to start with Joanne as the first department chair right out of the box. And then we're in the midst of recruiting a chief academic officer. MGH traditionally has not had a chief academic officer, a person tasked with setting strategy for research and education over the next couple of decades. We've traditionally managed our research with an operations leader, and our SVP of research is spectacular and great at that. That's what he's been tasked with. But his retirement, which I put off a couple of times, as some folks in this room know, but he came to me a year ago, September, on like my second week, and said, "By the way, I'm sure Peter Slavin told you that I want to retire." And I said, "No, he didn't." And he said, "Well, I want to retire." And I said, "No, you can't." And I used my my new president's chit. I used it not once, but twice. But the third time we agreed, and he'll retire in January. Part of why I wanted him to stay longer was I really wanted to get my arms around an understanding of our research programs. And I wanted to see both what was there, but also what was missing. And what was clear to me was that it's missing in what is an incredibly robust and powerful and talented and huge research program is a coordinated vision to ensure that in 25 years, when our time at leading MGH is done, that we're still the leading academic medical center, the leading hospital-based research program in the world. And I need help with setting that strategy. And I need someone whose job it is to wake up in the morning and think about that and go to sleep at night thinking about it, in addition to managing the nuts and bolts of compliance and the largest animal care program in the world. I didn't know that until I became hospital president, but that's true. So that's an exciting recruitment, and we're right in the middle of it. We have 5 candidates interviewing on Friday and Saturday of this week, and hope to narrow it down to 1 or 2 next week and get that person in place in January. So lots of leadership opportunities, transitions, lots of exciting things happening. but really none more exciting than the work that's being done in the Cancer Center with our incredible research program, clinical expansion, and trying to meet the needs of our community. And so I'll leave with that where I started. Cancer is my top priority. I know it's the top priority for everyone in this room. And together, we will make a difference. We will change the way patients experience their disease. We will, as Dave Ryan likes to say, we will be the place where people come expecting miracles, and we will deliver that increasingly for more and more patients with cancer. So thank you for joining us on this journey. Thank you for your support, and I'll turn it over to Daniel Haber, who you all know, my friend, my colleague, and the head of the Cancer Center. Daniel.
Thanks very much, David. Don't go away with a little model of the building. We want that building.
I'll leave it.
We'll have everybody sign it. It'll be, $1 to sign the new building. So welcome everyone to the second meeting of our Cancer Center Advisory Council. We had our first meeting in a really wonderful restaurant. And for the second one, we wanted to bring you to the research labs and get you a sense of what this is all about. In case you're wondering where we are, this was a torpedo factory during World War II, when the Navy Yard was actually a Navy Yard. It was then converted to luxury office space when the financial district was supposed to move to Charlestown, which it never did. And then it was converted to labs for MGH about 30 years ago. And I remember at the opening ceremony, a number of outsiders say, you're never going to fill this building, it's too large. And now 30 years later, there's no space in this building. There's another building of the same size on the main campus, the Simcus building. And as Marcella knows, we're trying to build more buildings because we're out of space. But basically, it's a fantastic building and we really love it here. So let me make just a few introductory remarks and then we launch into the program. First, I want to welcome our new attendees, Emily and Patrick Venanzi. Thank you very much for coming. Ashley and John Ranelli, again, thank you very much for coming. And then Kelly and Jason Krantz are here, but they're here virtually. We can't see you on the computer, but we know that you're in the ether. So hi, Keely and Jason, thank you for joining us. And we have some returning members, Dan Sherman, Belinda Tremere, Barbara and Marshall Tobins. And again, virtually, Arthur, Sandra, and Sarah Irving are also joining us virtually. So again, we can't see you, but hi, Arthur, Sandra, and Sarah, thanks for joining us. So at the first advisory council that we had, I was the one who was quarantining from COVID. So I was on a video looking by Zoom and speaking by Zoom. And this time we've set this up as a hybrid meeting. So again, those who are virtually can hopefully see and hear everything. And we'll reach out to them if they have questions during the meeting as well. In addition to our advisory meeting, advisory council, I want to thank today our board member, Carl Martinetti, who's also here virtually. And then our clinical director, Dr. David Ryan, our deputy clinical director, Dr. Effie Hochberg, our vice president for cancer services, Mara Bloom, and all of our clinical and departmental and development colleagues that you'll meet during the course of the day. So why are we here? What's the goal of our advisory council? It's essentially to support the mission of the MGH Cancer Center. And our mission, we define it very broadly. First of all, it includes generating fundamental knowledge and making discoveries that will impact the treatment of patients with cancer. Secondly, it involves providing the most advanced, the most comprehensive, and also the most compassionate clinical care for our patients and for their families. Third, it includes a true commitment to equity, diversity, and fairness, both for our staff and for our patients. And then lastly, it includes the training of a new generation of physicians and scientists to ensure ever and ever better outcomes for patients now and into the future. So we asked two things of our advisory council. First of all, help us secure philanthropic funds for these priorities. And this can take the form of building connections with other philanthropists, foundations, investors, business partnerships, in addition to making your own generous gifts, for which we're incredibly grateful to support our cancer center. And second, it's pleased to give us feedback on how we're doing in meeting our mission. Now, if you want to, you can tell us how to build a better T-cell. Marcella has an open inbox for all suggestions on her research program. But basically, we want your thoughts on how we're doing in meeting our mission and also in running what's a very large, complicated, and mission-focused enterprise. It's a big job to run this place. You all have expertise in business management and in how to make things viable. And that's one of the biggest challenges that we have. So again, we very, very much welcome your questions, your feedback, and your support. Since we have a few new council members, I thought I would just give you a little bit of background on the scale of the Cancer Center. We're a matrix cancer center at MGH, which means that we oversee basic research, translational research, medical oncology, and then we partner with all the other departments at the hospital who work on cancer, from surgery to radiation to pathology to psychiatry to diagnostic radiology, orthopedics. Everybody who has something to do with cancer works together with us in cancer care. So to give you a sense of scale of numbers, last year in 2021, we saw 24,000 patients newly diagnosed with cancer. We had 200,000 outpatient visits at our Yaqui Center, 60,000 chemotherapy treatments, and 10,000 inpatient admissions. All of these patients were seen by over 400 clinical faculty across all departments of MGH, all working in our Yaqui and our other areas within the Cancer Center. They're all grouped into 19 disease centers. Each disease center is a type of cancer, like sarcoma or brain tumors. And we have 15 specialty centers, which specialize in areas that are so complicated that you need to bring experts from different departments of the hospital. How do you deal with back pain or spine pain or other areas where you need to bring a surgeon, an anesthesiologist, a cardiologist together to solve the problem across disciplines? And that's what you can do in a general hospital. That's really, really hard to do in a cancer-focused institute or hospital. In terms of basic laboratory research, our Center for Cancer Research, our CCR, is based here in Charlestown. We have 50 lab-based faculty who study everything from how and why a cell decides to divide and become a cancer cell, all the way to how can you find molecular signatures of cancer cells in the blood for early diagnosis. We raise about $70 million in outside federal grants and foundation grants every year. In terms of clinical research, last year in 2021, we enrolled 3,300 patients on over 500 clinical trials. That's a big number for a general hospital like ours. But what's particularly important is that 40% of those were so-called phase 1 or first-in-human drugs. So those are the drugs that have never been tested before. And in the past, it was kind of random luck as to whether they work or not, but not anymore. Now, these are the drugs that are designed, they're specifically designed against a specific type of cancer with a specific kind of mutation. And basically, they come out of the box with a very, very high chance of promise against a specific cancer. And they're accessible in very, very few places. And we're one of the lucky places, in part because of who we are, in part because of the Tremere Center, which is where we focus on all of these, in part because we're based in Boston and all of biotech and pharma are right across the river, and that's where these drugs are all being made. So over the last 10 years, our Tremere Center has led or participated in a big way in 24 different drugs that got FDA approval for cancer. And again, that's an incredible record. So that's our shtick, is to take new drugs that have never been tested against cancers that are considered untreatable and see if they work. If they work, you get a phase 2, a phase 3, eventually it goes all the way to FDA. And the example that we gave you at our first meeting was about an antibody drug conjugate, which was developed here in partnership with our Tremere Center in phase 1, went to phase 2, went to phase 3, got FDA approval, and is now considered standard of care. And then again, that's something that we can do. In terms of the overall financials, now that the president is gone, I can tell you that the cancer service line generates, basically is $1.1 billion and generates a very significant part of the margin for the hospital. But as David has also told you, all U.S. hospitals right now are really struggling, in large part because of the rising cost of labor. So this has been a very tough year for the hospital. But thankfully, and thankfully to our colleagues here, development and our philanthropic campaign has gone very well. So in a typical year, we raise about $38 to $40 million in gifts, which go to all the specific causes throughout the entire cancer center. And then we have two major fundraising events, which are general to the cancer center. We have our center stage gala at the Museum of Fine Arts, which is really, really fun and cool. And last year we raised $2.2 million there. And then we have the Everyday Amazing Race on the Esplanade. So that has kids walking and running, adults running and walking as well. And we just raised $750,000 for that. And that again goes towards our priorities. So how do we set up priorities, our philanthropic priorities for the Cancer Center? The first thing is we really go for exceptional scientific and clinical promise. So that would be like the CAR-T program that you're going to hear about today. We also have other immunotherapy and targeted therapy approaches that are incredibly promising, and early cancer detection initiatives. The other way we set our priorities is really the need and impact of our patients, particularly in areas that are not well reimbursed or not well funded. And that includes supportive and compassionate care initiatives, diversity and equity initiatives, things that we need, that we value, but that not otherwise are very hard to support. And then, of course, there's the very large capital need shown by this beautiful little model there, the new clinical building, which is going to be absolutely outstanding with all private rooms, all integrated care, and will be the home of both outpatient and inpatient care for cancer. So today we're going to present you one of our shining lights, our CAR-T program with our shining faculty and scientists and clinicians. So what's a CAR-T? It's a bioengineered T cell. It's one of the immune cells from your own body, minding its own business. And Marcella and her friends will fish it out of your blood, engineering to make a drone out of it so it has a homing address, grow it up, and then give it back to the patient. So you take a cell that was just walking by, and you say, okay, you go kill that cancer cell once I've grown you up into billions. And it has had tremendous success, particularly in lymphomas and myelomas, a number of hematopoietic cancers, and hopefully in other cancers as well. There are only very few academic institutions that have the ability, have made the investment to actually be able to create cells like that. So in fact, there are a number of pharma companies that have certain products that are FDA approved, but the universe is huge. So just because there's 2 or 3 that you can buy, there's hundreds that you could make against different types of cancers. And that's the specific area for Marcella and her team. And in fact, from what I understand, there's only 12 institutions across the US that have the ability to design and test their own CAR-Ts made at home. And we're the only one that can do that in New England. So as you'll hear, the particular importance of CAR-T is the promise that academic centers can make more diverse CAR-T, more innovative CAR-Ts, more ingenuity in terms of how to define the whole universe of what can be treated with these therapies. So at MGH about 5 years ago, we had the good fortune to recruit Marcella Moss. She came from UPenn, from the lab of Carl June, who was one of the greatest pioneers in this technology. And over 5 years, she's trained a whole team of scientists and physicians who you'll meet today. And particularly, she's also trained Dr. Matt Frigault, who's our clinical director and who's going to lead off this discussion. We've become a major international referral center for CAR-T. So to date, we've actually treated 359 patients with CAR-Ts. Some of these are off-the-shelf commercial, some of these are research partnerships with biotech and pharma, and some of them are homemade in Marcella's lab and being tested here for the first time. So you'll hear from Marcella about what the future looks like in cancer, in other diseases, everything that can be done by engineering T cells. And finally, as a vote of confidence, our parent system, which is now called Mass General Brigham, has decided that cell and gene therapy is going to be the future and the place where they're going to invest tremendous amounts of resources. And they've appointed Marcella as the leader of the cell therapy program for Mass General Brigham. So she's kind of our friend here, but she's going to be ruling supreme across the entire system. So with that, let's start the program, and we're going to start with a clinical discussion led by Dr. Matt Frigault and his patient, Jillian Dashko. If I can invite you to the podium, or to the chairs.
I apologize, I'm losing my voice, so I'll keep this as loud as I can. But so I'm Matt. Hello, everyone. I'm the clinical director of the cell therapy program. I kind of fell in love with CAR T cells over a decade ago when I was a graduate student, and was just kind of blown away by the Frankenstein kind of science that was going into these things. And was actually told my first year that it will never go anywhere. It's too much of a boutique thing, and not many people can do it. And now having treated multiple patients like Jillian, it's kind of rewarding to be able to do that. So just to reiterate what CAR T cells are and why they're important is that, we know with immunotherapies that T cells, our immune system, pretty much fight off cancer. And I think I gave you the spiel many, many years ago when I was discussing the trial with you. On a daily basis, it's fighting off cancer in many cases, or in few cases that are unfortunately end up becoming cancer, it misses. And so that's where these other new pembrolizumab, nivolumab, ipilimumab, right? Those for solid tumors really came about and really changed how we treat metastatic cancer. Well, when it comes to therapies like CAR T cells, it's kind of a whole new level where you're taking a natural immune response, the generals, the snipers, as I call them, and you're genetically modifying them, giving them new instructions and redirecting them against a target that would otherwise not be targeted. In the case of Jillian, we were targeting something called CD19. There may be a lot of car analogies in terms of traffic and stop signs and whatnot, but I say CD19 is kind of like the exit sign on the highway at night, where if you're driving down the highway and you don't see it, you really don't know where you are. You could miss your exit and then three exits down go, oh my God, there's exit 19. But that's the exit sign for B cells. And so what that allows is basically says, I'm a B cell, and it's on good B cells and it's also on bad B cells, lymphoma. Now, Jillian had a very rare kind of lymphoma called primary CNS lymphoma. So it's a lymphoma that starts and stays within the central nervous system. And we'll go in a second about kind of your initial treatment, which wasn't even for primary CNS lymphoma. Just to tell you how rare it was, she was actually misdiagnosed initially. She was getting treated for something completely different that was more immune mediated, until she kind of made her way around and was fortunate enough to finally get a diagnosis. But the reason why this is so important are that we have CAR T cells for very many types of lymphoma now, from mantle cell lymphoma, follicular lymphoma, diffuse large B cell lymphoma. But every single clinical trial that has ever happened, even probably now, has pretty much excluded primary CNS lymphoma. They're too rare, it's too high risk, and everyone was worried about some of these neurotoxicity side effects that we see. So patients can get really sick, have low blood pressure, but they can always have swelling in the brain, confusion, and things that happen. So we ended up developing and working with FDA and some companies to develop the very first primary CNS lymphoma clinical trial. It took about, a year or two of really pushing this thing along, but we eventually got it up and running. And that's how I got to meet Jillian now, probably almost 3 years or more ago, and kind of where our story began. I pointed out, I think a little bit earlier, that this is Jillian's first time in Boston, where she's not coming to visit to get a brain MRI.
Yes.
So we're celebrating that, and that she's enjoying some, what'd you say, bougie restaurants.
Yes.
And having some fun in Boston. But we'll just step back to kind of, I want Jillian to tell her story, because I think it's really powerful. So you were initially diagnosed about 3+ years ago.
Yes.
And at that point, what were you being told?
Before my
Correct
diagnosis. I was told that I had a rare form of MS, ADEM. Yeah. And they were treating me for MS. But I wasn't getting better. They were band-aiding it. I was good enough to go home and be out of the hospital, but I kept coming back with different symptoms, different problems.
So then you made your way to, I believe it was the University of Pennsylvania?
Yes. I went to University of Pennsylvania, where they ultimately did a brain biopsy, and that's where I got the correct diagnosis.
And so then you were started on first-line therapy, which is basically high-dose chemo.
Yes.
And how'd that go?
Methotrexate was the chemo, and it went well for 4 months. I went back for my 5th month, and they do the MRI before everything. And at that appointment, they told me it's stopped working, or a doctor did tell me that my cancer was smart enough to know, okay, this is trying to hurt me, and stopped it. I don't know.
So that was scary
That was very scary.
because that's our mainstay.
Hmm.
And then where did they send you?
They sent me to the NIH for a clinical trial that was going on, TEDDR, and we started that, and it went well for 4 months. And then the 5th month, same thing. I went MRI, it's not working. And they didn't know what to do. And that was not the first time I see confusion with doctors, but I think they knew that there was not much more I could continue to do. But the one doctor did say, there's a doctor in Boston who is starting this trial, and I'm going to see if I could get you in there. And he called Matt.
I remember getting an email from Mark and hearing about this young woman who was failing high-dose methotrexate, failing TEDDR. And they have this, we all like fancy acronyms in medicine to sound cool. And that was when you started Viper.
Yes.
And so she started this new experimental therapy on top of the other experimental therapy, which is a combination of basically every inhibitor you can think of for B cells. And you initially had a partial response. And this is while we were trying to buy time to get Jillian up here. And it was around that time, I think, that we first met. And you came up to Boston, and we basically get everything lined up so that I think we had your visit, your labs, and had you collected. So we had to collect the T cells to manufacture them within about 3 or 4 days. So we had lined everything up to make it quick.
Yes.
And I remember you were still on steroids at the time, and we were figuring out if that was important. And we said, okay, so go back to the NIH, and we're going to give you another cycle of Viper because we need some time for these cells to get manufactured.
Yes.
And I remember getting a phone call. You got another cycle. I remember getting a phone call from you, and what happened?
I started having weakness on my left side. Every time I stood up to walk, my ankle would roll and I would fall. And obviously, I knew there was something going on. So I called Matt and I said, I don't know what to do. What should I do?
And I remember saying, just get in a car and drive here.
Yeah.
I'm like, we're like 2 weeks away from these cells. Just get in a car, whatever you can do, just get up here. We'll admit you, we'll take care of it.
Yep.
And I remember we started high-dose steroids, and we're basically buying time for these cells to come back and get manufactured. And I remember kind of seeing you in the hospital, and then finally we got the cells. We were able to start the chemotherapy, which is very low doses to, again, another car analogy, open up the highway. You can be in a Ford Pinto or Ferrari on 993 during rush hour. You're not going anywhere. So the LDC or the chemotherapy removes the other cars from the highway. And so you got your chemotherapy, and then we gave your cells, and then I had to fly out like 3 or 4 days later to go give a talk at the VA. And you had started to have some fevers, and things were okay, right? And I remember at that point, you had some pretty significant left-sided weakness.
Yes.
And that was a little bit nervous. And I think we were getting MRIs like every 3 days because we were really, really worried about Jillian. We had you on steroids and other therapies. And then something really scary happened. And I think Kyle probably remembers this a lot more than Jillian does. But then I think you lost all ability to move the left side of your body.
Yes.
No movement whatsoever. And then you kind of fell asleep and you went into a coma. And that was a mixture of, looking back, and thankfully we can do the labs and look at the research samples, understand that that was a mixture of your disease, but also the CAR T cells starting to work, where they were causing inflammation around your tumor as they were attacking it. But your tumor was already causing so much inflammation that you really didn't have that much more room to expand to. And so we gave some medications, we tried some maneuvers and said, hey, let's try this little low dose radiation just to kind of pump the brakes. And Jillian woke up. And what was that like?
Confusing. Scary. But I had Matt, an amazing team that reassured me all along the way. It was during the beginning of COVID. So in the beginning I had family, but then they couldn't come anymore. And that was hard.
Yeah, and you actually you basically have been on that clinical trial, so it's 2 years long, throughout the entire pandemic. She has been flying back and forth. We've been crossing our fingers, getting vaccines, giving monoclonals, doing everything we possibly can
Mhm.
back and forth to come get treatment for this thing that no one else really wanted to touch at the moment.
Yeah.
But I remember there's one cool story. I remember I got back right when all this was kind of really hitting its peak, and you were just starting to wake up. And I remember you being really down. And I remember being next to your bed, and what happened?
I lost my complete left side movement, and Matt picked up my arm, and well, you did it a couple days. He would say, try and move your fingers. Nothing happened. But the one day he said, okay, try and move your finger. And I was able to move my finger. And I think we both burst into tears. Yeah.
So that was, that was a scary moment going to potentially happy moment.
Mhm.
But I think the hard thing for you is that, all these therapies and disease kind of took its toll to some degree, right?
Yeah.
And I know we've talked, but it was a long road after that.
Oh, yeah.
It took you probably, I say, a solid year to get back to where you were, a lot of physical therapy and other things. What has that been like?
Difficult. I still do physical and occupational therapy. And I think I'm probably going to do that for a very long time. Maybe forever. I'm comfortable doing it because I learned, I'm worried about my balance, and they, make sure that I'm on the right path and doing the right things and learning things. I'm unable to type as fast as I used to before because I'm down to one hand. A lot has changed in my life, but I'll take this over what could have been.
Well, you also have Brady.
I have a son. He's 15. Yeah.
Yeah, I remember he's grown up a lot in
Mhm.
that time, like doubled in size, probably.
Yeah.
Like,
I remember I came home from the hospital, and the first words that came out of his mouth, I don't even know what they were. I just said, "Oh my God, your voice is deep." Yeah. I missed that whole transition.
so that it took some time. But,
Yeah.
he's now playing football, right?
Yes.
He's back home.
Yep.
And so I think, an important thing to kind of point out is that there are a lot of signals, a lot of signs for Jillian that traditional stuff wasn't going to work, right? We had done the methotrexate, we had done the TED-ER, we had done the and we were really just buying time at that point. And so it became a big push for us to really notify people that you need to get on this sooner for patients like Jillian.
Mhm.
And I think earlier treatment would have been really important.
Yeah, I agree.
But we finally hit our 2-year mark. Was it 6 months ago?
In February.
Yeah. So we hit the 2-year mark, and that was a big celebration, but also a lot of anxiety because
When I come for tests, I'm getting out of the MRI machine and I'm texting him. Did you read my MRI? But it's stressful coming here. This is the first time it wasn't stressful. So thank you, everyone. But yeah.
And so I can say, after all was said and done in terms of kind of what happened, we published a study. We were able to treat 12 patients, which for the price tag of these CAR T cells, you can imagine that's not an easy thing to do for things that are off label. And I haven't really updated you on this. So we're working with organizations like NCCN, which help put out standard criteria. And I learned a little loophole. If it's recommended by NCCN, whether it's on label for FDA or not, insurances will pay for it. And so we're making a big advocacy push for NCCN to recommend it for primary CNS to get patients like Jillian access. But because of Jillian's participation as well, we've probably been able to treat another 10 or 15 primary CNS lymphoma patients, because I can now go to insurance companies and make an argument for why this works and for why they need to support it and for paying for it. And so that's because you took a risk. You're one of our first patients. We had no idea what was going to happen, and she took a big chance, and here we are today.
Yeah.
So I think you just built a new house. Right?
Yes, we did.
We went from being all scared to getting pictures of her new finishes that she was getting done. So it's been a meaningful experience for us, and I'm not sure if you have anything else to say in terms of take homes or big messages for folks. Not to put you on the spot, I'm sorry.
No, my first meeting with Matt was the first time that I met with a doctor after my diagnosis, and I felt positive. He did say, we're gonna cure you.
said that.
You said something around that.
I'm usually
We're
very-
looking for a cure. We're not
Oh,
looking
okay.
for a treatment. We're
I was
looking
like, we're
for
going
a
for a
cure.
home run here. We're not.
Yes.
We're not going
We're
for 3
look-
months. We're going.
yeah.
If this is going to work, I want
Yeah.
to. So I'm like,
Did
come
I
on.
say that? And I felt positive and comfortable here. Yeah. So
And we were actually able to treat patients now, I think, from Pennsylvania. And you met another patient that you referred up to us through a Facebook group.
yep.
And so there's this little network of primary citizen lymphoma patients on Facebook that things got buzzworded from Florida, from California, from Chicago. And so patients were flying in the middle of COVID, like brand new patients, getting on planes, on chemotherapy, no B cells, very immunocompromised in a scary time. And we were just saying, get here and we'll do everything we possibly can. And we did that during the middle of COVID. So I think that should give me some extra points on the publication, but I'm just happy to see people like Jillian doing really well and having others like yourself continuing to do well.
Thank you.
Thank you very much. Please stay here. We're going to have the panel join you. Effie is the chair of our panel. If you'll bring your speakers with you. Then we'll have a panel discussion and then an open discussion with everybody on the panel.
I'm Effie Hochberg. Very nice to meet all of you. I'm the Deputy Clinical Director of the Cancer Center, and it's such a pleasure to work with incredibly smart people and to hear that incredibly moving story. It was particularly personally meaningful for me. My father was a neuro-oncologist here for 40 years who worked on designing the first high-dose methotrexate trials. And to kind of see another stage in the development of therapy for this disease come through our institutions personally generates a lot of pride in me for our institution. But thank you very much for speaking. So I wanted to start by asking all of you to think as we're talking about Mass General Hospital, what resonates with you about the care we provide, what resonates with you about the research that we are trying to provide. What do you see as exciting in what we are saying about the care that we're providing, about the research? What do you see as places where there are deficits in what we're talking about, the kind of care we provide? How do we make our care touch a broader group of people? But we'd love to hear your questions at the end. I'll kind of moderate those questions. I'll go through some of the individuals here, and then we'll talk about some of your questions if you have them. So maybe I'll start with Marcella and Matt. So maybe, Matt, if you could go through some of the places where we do have CAR T-cell therapy, and then Marcella, if you can talk a bit about your interest in the future of CAR T-cell therapy. What are the targets? What are the diseases? What's the research you're most interested in?
Hello, can you hear me? Great. So, right now we have, initially the first approval for CAR T cells was in pediatric and young adult B-ALL. And that was a really exciting time. These was not only the first cell therapy approved, it was actually the first gene therapy approved on top of that. So it was kind of a really exciting to see that come online. And then very quickly thereafter, approvals for diffuse large B-cell lymphoma, multiple products, mantle cell lymphoma, recently had approvals for follicular lymphoma, adult leukemia was recently approved. We have approvals in multiple myeloma as well. And we're now looking for things like melanoma. So solid tumor cellular therapies are probably going to be approved within the next 12 to 16 months. So we're prepping that whole infrastructure. And I can tell you that of all the indications I just mentioned, we have treated every patient with every product, which is an exciting thing to have access to. Not many places are taking everything on like that with the risk, but we've used every product in every instance for every type of patient.
So when you look at the sort of CAR T field, there's so much going on, it can be a little bit overwhelming. So to sort of distill it down, I would say we're focusing on 4 things. One is to try to decrease the toxicity, the side effects, and that would enable outpatient treatment, like a full-scale outpatient treatment where people don't necessarily have to be either in the hospital or within 30 minutes of the hospital. And so we know that the immune system is highly druggable. And so we're trying to use different therapies that are already out there, but maybe have been tested in a different disease like autoimmune disease. And testing those in the setting of CAR T to see if we can make it more tolerable. And so together with Matt, we've already developed 2 clinical trials testing this. I would say we're still trying. We have some, I wouldn't say not total misses, but not a complete win. And so what we really go for, Matt and I are pretty ambitious that way, we want the full win. We want to make it so that we preserve all the anti-tumor effects, but then have it be like smooth sailing. And so we're just actually proposing, right before this meeting, we were in a call proposing a new trial with a company that has a drug that we think would be really like get at the root cause of the side effects. So that's number one, is trying to make it more accessible, more tolerable, easier for the patients to get through. Number two is we want to look for new targets, because some of our patients with lymphoma, with myeloma, their disease comes back. And sometimes you need another exit sign. You need another route to get to the destination. And so some of the things that we're doing in my lab, and you'll meet some of those folks later on, are developing new targets. And so we go all the way from like testing it in a dish to a mouse, and then we try to train. If it looks promising, then we try to take it to people. So toxicity management, like side effect profile, new targets, solid tumor. And so solid tumor is the vast majority of cancers, but it hasn't really been as easy to target them. And that's for a number of different reasons. Some of it is just how we diagnose cancer. The same things that we use to diagnose leukemias and lymphomas are kind of those are the exit signs. That's what we use to figure out how we should target it. So it all aligns really well. Whereas diagnosing a solid tumor, you use completely different formats. It's a lot based on how it looks under a microscope. That doesn't necessarily tell us how to get the CAR T to it. And so we're trying to kind of figure both of those things out at the same time. And then finally, we want to make it just more accessible to more patients, not just from a side effect profile, but like the whole shebang, the whole manufacturing process, the cost, the payers, the insurance. There's a lot of moving pieces. And so we want to be at the forefront of making this sort of accessible and equitable to more patients.
Maybe I can pull in something that Jillian, you said earlier about how sick you had become and the need to get some therapy to keep you in remission for a while because of the time that it took. How long was it from when you first met Matt until you got the cells? Do you know about how long that was?
I think, did we meet in January?
Yeah, it was quick.
It was quick, January, and then February
Scary.
13th maybe was when I got my cells.
But I think you could hear in the story about how ill you could have become in that time period. And maybe Marcel, if you could say one or two more words about our ability to try to improve that process of making cells available more quickly.
Yeah, this is definitely a group effort. There's a lot of moving pieces that have to come together. Matt mentioned about aligning, collecting your cells, seeing your visit, getting your MRI, collecting your cells. That's hard to do, especially at large scale. And so trying to shorten that time for where the CAR Ts are getting made so that you don't have to go a longer period of time getting a therapy that's not really doing the job is something that we're aiming for. So just to give you one quick example, in our trials, we use a 7-day manufacturing process, whereas the first ones and the ones that are commercially available are 14-day. So even though it's shaving off a week, that's every week can kind of count in this scenario. So trying to make that even shorter. And there's efforts that we and others are also taking part. We're trying to get it down to like 2 or 3 days. Which would again make it a much more like faster turnaround. Maybe you could do the whole thing in 1 or 2 visits instead of having like a 6-week time in between.
So I wanted to turn now to Sarah Cobb and to Sarah Noyes to talk a bit about the other members of the team and how much of a group effort it is to get patients on these clinical trials and to move the trials forward. Maybe Sarah, if you could start by introducing yourself and then talking a bit about what it takes to get patients here initially for the first trial, and then what it took to get that trial up and moving.
Sure. So, hi, I'm Sarah Cobb, and I feel like there's like this continuity because actually I worked with your dad as a young nurse. Yes, I did on this first CNS, the high dose methotrexate, BCNU, fluosol trials, and the brain cancer patients always have like a special place in my heart. And that's what I tell Matt. I said, when we were working on this trial, I'm like, I'm so glad we're working on primary CNS because, there was just not a lot of options. So it's so great to see you here.
Thank you.
So my job is to do that whole organization part. An email, sometimes like literally an email will come in, Matt will shoot me, he'll CC me on an email. And it's from another doctor saying, hey, I have this patient, can you see him? So then the ball starts to roll. And a lot happens before a patient arrives for their visit. So because of the time delay, I start to get everything all set up so that if they sign consent, especially if they're traveling, we're ready to collect. So what will happen is, and collection is a morning in an apheresis lab, and it's a procedure, and sometimes they need a line, so there's another procedure. So like, for example, we had a last minute patient we need to do on Monday, and it was a lot of pushing on Thursday to get interventional radiology, to get the apheresis lab, to get the patient to everything had to align correctly. And sometimes it's really important. Sometimes we have time and sometimes we don't. So I have to say that I'm proud to work at MGH because I had multiple departments that really emptied their schedule and worked to get these patients in because they recognize the patients are sick and they really need the care. So that's a good thing. I'm happy to work at an institution where people are like, no, can't do that. They're really like, what, we'll try, give us some time. So the patient, they need a lot of scheduling to come in and see the doctor. And so the doctor the nurse navigator, myself and Debbie sort of like make the plan. And then we say to the physician, do you like this? This is what we've got, what needs to be tweaked, and they're like, great, so, patient has multiple visits that they come in, they have a consult, they may have to come back for a collection, they have to come back. They are seen by their referring oncologist for bridging therapy that we call, because we have that 3 to 6 week time frame between the time of collection and the time they get their cells. And then they come back again for clearance, then they come back for chemo, and then they're admitted to the hospital. So it's a lot of resources and it's a lot of strain on the patient and the support. The patient needs to find their own support team too, that they can't be alone. They need somebody to bring them in. That's a big, sometimes it's a big challenge. And we work with social work too, to try to get, some patients have a great support system and some patients have very little. And very little resources. And it's trying to we never want to turn a patient down because they don't have what they need. But it can be hard. They could be sort of alone, and we think creatively, like who can come with you, and then the manufacturers have plans that help with lodging around when they're at MGH because they have to stay here for a month. But some patients don't either qualify, or sometimes they live close, but they have just as great challenges coming from Dorchester to Boston multiple times. It can be as much of a challenge as coming from Maine. So those are some of the things that
Hopefully you get the sense of how much effort is required to really provide the care. It's not just the science, it's the human support of the individual involved and trying to make sure our system can support them. So I want to turn over to Sarah Noyes and talk a bit about the extent of our current program and what it takes to keep all of these trials Matt is mentioning up and running.
Hi, I'm Sarah. I am an inpatient nurse on the floor that we actually give the cells. So I take care of the patient's bedside with the attendings and with the team. I would say I worked on the research team outpatient before I went to the inpatient nursing side, and it takes a village. There's all of the other departments, so many moving pieces to kind of bring it all together. And from my perspective, from nursing at the bedside, these patients can be very sick. Some breeze right through, some it's great, and then some really require some almost ICU acuity. As much as, Matt loves getting my 3 a.m. pages saying we've got a fever of 104, blood pressure's dropping, confused, and all these things. It takes a lot of education. We are constantly learning more things, constantly asking, this is a new product, what can I expect of this? what goes into this, what do we do here. But it is pretty exceptional getting to see patients like Jill and the new studies coming down the pipeline and things we usually wouldn't have other treatment options for is really, really incredible. We've come such a long way. I remember, I think it was early 2016, we treated our first cell therapy patient. We were all kind of sitting waiting for something to happen. And, a few months later, the patient was able to meet her grandson, and it was really, really special. So we've come a long way. The program has really exploded, but it takes, it's not just nursing, the outpatient, inpatient, it's the cell lab manufacturing lab, but then the cell lab on the site, and then the blood transfusion services. There's so many moving parts to really bring it all together, but it's pretty incredible what we've accomplished so far.
Thanks very much. Appreciate it. It really gives us a perspective of the intensity of work that's required on the inpatient side when you talk about the side effects that you were experiencing and the loss of function in your hand. There's Matt gets to come by and experience the good news, but I think there's a nurse by your bedside every day for probably in the hospital for a couple of weeks. Maybe we'll switch to a broader scale perspective. I'll call on my colleague Dave to talk a bit about, first introduce yourself. I think everyone knows you, but maybe not. And then talk a bit about the ways in which the Cancer Center is focused on CAR T-cell therapy as one of our paths forward into the future.
Sure. So, hi everybody. I'm Dave Ryan, and I run ultimately with the whole team, the clinical operations of the Cancer Center. And so, I think I couldn't be more proud of the group and of the patients that come to the MGH for this therapy. And the way that we look at it from a leadership perspective is how can we best set up the structures that allow the physicians and nurses and ultimately patients to thrive with these really difficult cancers to treat. And the structures that we've been able to set up over the last several years incorporate not only the best standard of care that money can buy, but the best research that money can buy, and also the best supportive care that money can buy. And it's that combination, those three legs of the chair, that help patients get through these very, very difficult times and ultimately leave the field better off than how we found it, which is really what we want to do so that people can go on and live incredible, wonderful lives. And so the structures that we've put in place, we have the examples and we've set them up over the years and built on them. We're actually using our Tamir model to set up the CAR T infrastructure. And what do I mean by that? So the Tamir model is where we do all of the investigational chemotherapy clinical trials. Every disease center that Daniel mentioned has members in their disease center that have a foot in the disease center and a foot in Tamir. So they know exactly what's coming out of the mouse labs, the animal labs, the cell culture labs, what is targeting those particular cancers. And the first time they go into people, they actually have an idea, is that going to work in my tumor type for my colleagues back in the GI Cancer Center or the breast center or the neuro-oncology center? Same thing now is true with this revolution. Every time that we think about immunotherapy, particularly engineering a T cell, we want our individual physicians and nurses to think about, is that going to be meaningful to my patient with stomach cancer or to my patient with lung cancer, not just lymphoma, not just myeloma, not just leukemia. And so we are developing a cellular therapy organization, a team that has members embedded within the team, but also embedded with all the disease centers throughout the entire hospital, throughout the entire cancer center, I should say. And so as we develop the infrastructure, we want people to be able to thrive and to develop the new, better therapies that we have. And so everything about cell therapy is complicated. Everything about it costs more money, needs more people, is that much more expansive in terms of the structures that it needs. But you see the results. The results are unmistakable. And so as we think about this in terms of leadership, we need infrastructure for the standard of care. So you heard about the apheresis lab. You heard about the cell therapy pharmacy. So who's processing those cells in a sterile fashion after they're collected, and then they go out to a company or to an academic institution to manufacture, and then they come back. They have to be processed sterilely, and then they have to be put into that patient. Who's creating the infrastructure for inpatient nursing, for inpatient management? Who's creating the pathways so that when somebody all of a sudden goes into a coma, that they're managed at 3 a.m., that the neuroradiologist is called, that the neurosurgeon is called, that the team in the ICU is made aware. All of that infrastructure on the standard of care has to be developed. And then finally, the research component needs to be developed. Every single patient who went on that CNS lymphoma trial that Matt and Eben and others put together, it was a dream, but they went somewhere and said, we need to do this. It's the right thing for patients. Just the data management cost at least $30,000 per patient. Just the data management. None of that comes from industry. None of that, comes from other places than philanthropy and the help that we get from the grateful folks in the MGH community. And so all of this is part of the structure that goes in to making MGH the greatest place on the planet to be a patient.
Whenever you say greatest place on the planet, we always stop and applaud.
So maybe I'll take one more second and extend off the point you made, Dave, about philanthropy, and really ask Marcella and Matt if you could talk for one moment about the impact that philanthropy has had for both of you on the scientific side, on the clinical side, and the work you've been able to begin to accomplish.
I mean, I feel like the word gets used a lot, but boy, is it true here. It's just transformative. We couldn't do half the things we do, or maybe even more than that, without generous donations. There are grants from the NCI and other organisms and foundations that will fund preclinical research. Like if I want to test something new in a mouse and I have a hypothesis, we can get funding for that. And I don't want to say it's small dollar amounts, but the amounts of money that it costs to treat a mouse are peanuts compared to what it costs to try to bring something to a patient. So there are mechanisms in the US to fund that sort of research. There's also mechanisms to treat patients with standard of care. We bill insurance, and I'm the least qualified here to talk about exactly how that whole system works, but it generally works. It's the stuff in between. It's called the Valley of Death for a reason. And in some ways, the biotech companies around Boston and other biotechs have filled that need, but they take a long time, and sometimes their time horizon and motivations don't exactly match what our patients need. So for example, because they have to build a whole company around it, it may take them a lot longer to get to a phase 1 clinical trial because they have to hire a lot of people. They spend a lot of money doing so. Their cash runways are 3 to 5 years, and sometimes it takes longer than that. And they're not as motivated to treat diseases that don't have a big market potential, that they're not going to IPO or treat large numbers of patients on the. So for example, I've been very interested in treating patients with glioblastoma. That's a disease that most biotech companies aren't going after because they don't see a potential business proposition there. But if we make a transformative impact, it will come. I think there's just a different kind of appetites for risk, and some developing a drug or a therapeutic in some diseases entails more risk than others. And so we have been really, and I'm just so grateful for all the effort that everyone here has made to put it in our hands a little bit to figure out what we have. Great doctors who, when the patients come to Mass General, I think they can identify where the needs are, and with the right science and the right motivations and relatively not the kinds of dollars that it takes a biotech company, but still transformative gifts for us in the under $10 million range, we can still treat 3 to 6 patients and really get a sense as to whether a drug is going to work. And so philanthropy is just absolutely key for us to be able to do this kind of work.
I think my microphone's on. So, just to reiterate all of that, not to use the word transformative too many times, but it truly is, whether it's the patient level, helping with hotels through fundraisers and philanthropic donations during travel in the middle of a pandemic, all the way to, Sarah, I can't say enough how amazing she is at her job and all the moving pieces she has to track, but there's no billing code for that, right? There's no way that I can charge an insurance company for the 3 hours Sarah spends trying to get a line lined up for an apheresis date for a manufacturing date. Like those are things that just, I will say, I remember, you had to take a risk, and we went in and we said we're going to do it and we're going to do it right. And those and the philanthropic donations and whatnot help support those types of risks that I think are really key. But now we're at a place where I know we were saying 300 and something patients treated. I think we had about 137 infusions just last year. I was looking at the final numbers. So we're exceeding our expectations, but it's just getting more complicated. So for us to keep pushing and pushing and expanding, we need those types of investments to just build the infrastructure so patients can get here, patients can get treated, and we can do it fast, efficient, and we can do it the right way.
So let's open it up for questions. Again, our panel will stay here. And any thoughts, questions, comments, advisors, or 3 to 5 years from now?
What are the 3 things that you need to achieve that success?
It was the question too.
We'll have. Yeah, the question is, in 3 to 5 years, how would you define success?
I think success for me at the looking at the broad MGH level is that we have established a CAR T service that is allowing young people coming up from the bottom, if you will, and we attract the best and the brightest young people from all over the planet. And Matt Fergalt was a fellow just 10 short years ago, and there's a whole bunch of fellows coming up through the system. So establishing a structure where MGH is considered the best place to implement cellular therapy in the world. That's number one. Number two, I would say success is defined by at least 3 products coming out of our internal cellular therapy labs that are making a difference in patients with cancers that have no ability to be cured today, so that we have more patients who are sitting up here saying, look, I'm building a house, right? And then three, I want us to introduce cell therapy into the solid tumor patient and crack the code that is keeping these T cells away from solid tumors. And what we mean by solid tumor, just to put it into English, breast cancer, lung cancer, colon cancer, pancreatic cancer, ovarian cancer. And I think we're gonna get there, and we're gonna get there because this technology has the ability to not just target a cell against the cancer, but can deliver almost any cocktail of proteins and mRNA directly to the tumor microenvironment. So that's how I would define success.
I like your definition. I was going to say open 5 trials in the next 5 years, and hopefully at least 3 of them result in patient stories like Jill's. I mean, that's makes me cry. I want to see more of that, and I want to see it in different contexts, and I just want to scream it from the rooftops kind of thing. That will be success.
So I think everyone already knows I want the home run. Oh, sorry.
Time for the question.
Yeah, we'll let it go. We can talk after.
Wondering, is manufacturing at all a possibility, doing it yourself rather than sending out?
Can you make it so?
I mean, President Biden is wanting to build manufacturing plants.
Marcel, just repeat the question first.
Yeah. So Belinda Tamir is asking about, is it possible to develop manufacturing here? And I would say absolutely. Cell manufacturing is well within the scope of what academic centers have traditionally done. It has gotten a little bit more complicated as it's gotten more and more commercial. Like the regulations are a little bit more stringent now, but it is completely feasible. The challenges are really around building it and then staffing and operating it. It's the jobs to do that are not traditionally academic jobs, and so finding ways to operate it, maintain it, and have like not just a revolving door of people coming in for training and then exiting, I think we have to think of models that will make that work. But I'm also really interested in the possibility of what we call point of care or distributed manufacturing, where a patient comes to the hospital, their cells are collected and they're manufactured right there. And I think that does a couple of things. It shaves a lot of time off. It shaves a lot of operational logistics off, which are complicated. You can't have the wrong patient get the wrong cells. And so there's a lot of effort to make sure that there's a chain of custody, chain of identity, all of these regulations around it. So if you could do it right there, it would really be transformative in terms of the turnaround time and the safety. And I think it would also bring down costs quite a bit. To have multiple centers. We're actually in talks with a couple of other academic centers to do some comparisons across centers where we manufacture ours and they manufacture theirs using the same protocol, and we can do some comparisons. It's in very early phases, but right now we're doing our manufacturing with our collaborators at the Dana-Farber. They've invested in a large manufacturing facility there, and it's part of the Dana-Farber Harvard Cancer Center. But I think in the future, it would be very useful to have an MGH manufacturing facility.
Questions? Let me ask one. When you make the transition from a really, really difficult, expensive treatment that few people can get to something that's affordable and accessible to all, can you name the few bottlenecks or the few things that have to change for that to happen?
Just repeat the question.
Sure. Daniel was asking about what some of the bottlenecks are from going from a sort of proof of concept in a small trial to something that's equitable and accessible on a larger scale. It's a little bit complicated. There's like a regulatory environment about the FDA regulates when you try to sell a drug. And so you have to, hospitals haven't generally been in the business of marketing drugs, and that's a regulated process, but you can treat your own patients through different processes. I actually have a fellow in the Harvard Regulatory Sciences in our group who is investigating this and writing papers about it with FDA. So the one is the regulation part, and then the other is just sort of the scalability of the operation. And I think that's also where treating our own patients in our own hospitals, we do complicated surgeries, we do complicated, in vitro fertilization used to be very complicated as well. And it's become much more standard and much more feasible. I think this is very similar in that way. And so it's eminently feasible, but you have to align all the stakeholders to make it happen.
This is sort of more to Daniel's point and a little bit of what. Sorry, Matt, you talked a little bit about the advocacy needed to the recommending body to get the insurance. Can you talk just in a little bit more detail about what that advocacy looks like? And is that a space that philanthropy can ever help to resource to expedite?
Really good question. In general, as Marcelo was saying, this is a really highly regulated space. And so we can present these data to the FDA and they can look at it and say, well, we want 100 patients over 10 different centers. And that's just not really feasible. Especially for rare diseases, orphan diseases, diseases with unmet need, any kind of philanthropic funding that supports the research that explains the rationale to do this trial, right? So like if we didn't have the basic understanding of toxicity, where we said, and just to kind of make it clear, we didn't see increased toxicity over any other trial that we've done for primary CNS. So all the concerns about the neurotoxicity really didn't pan out to the larger degree across the study. But that took scientific investigation to understand mechanisms, retrospective studies, correlative studies to build a knowledge base, to then go make an argument, to then do the study, to then go to those types of bodies and organizations. And so, a lot of the time is I'm just emailing the head of the NCCN board or calling friends and making connections and sending drafts of papers, and just getting people who have the right mission and want and desire to review it in a really fair way to say, is this something that should be more accessible to folks? There's probably a much longer answer to that. But I think, just having the evidence in the first place is probably where we're lacking in a lot of rare diseases currently.
Can I add one? Oh, sorry. Can I add one thing just in response to your question? Is that one thing that's also unique about cell therapy and being developed here is that we actually generate composition of matter IP. And so unlike when we develop a drug in combination or in collaboration with a company where we've maybe our scientists have identified a target, and then it goes out to a company and they develop the drug, and then it comes back to us as here's a drug you can use for your patients, which is also a good thing. Here, we actually own the composition of matter IP. So if we have a big win, we can license that and it can get commercialized through a biotech or a big pharma company. In fact, all of the CAR Ts that are currently on the market came out of academic labs. Not a single one of them was fully invented in a company. So NCI has developed two, Seattle, Hutch, like lots of different places have, all of the CAR Ts have come from first in human studies that were done entirely in academic settings. So if you have something that is, when you own the IP, it also brings in dollars to the institution and royalties and milestones and all of those things. So that's another possibility.
So in the interest of time, I'm going to thank the panel. We'll have time for more questions during the lab tours. And then our last kind of showcase here is going to be Marcella giving us a brief thought about the future.
So I think I've made some of these points during the panel discussion, but I just had a couple of slides that I was going to tell you about in just very succinct terms, like what are we doing and why? So I'm an immunologist by training. I study the immune system. And one of the reasons I'm so excited about the immune system is because it's involved in so many different kinds of diseases. So it protects us from infection. It can be incredibly powerful that way. It's also involved in many diseases like lupus, rheumatoid arthritis. And because it's so powerful and it's also so druggable, we can redirect it in ways that it didn't even really evolve to do, which is to like treat cancer. And even potentially cure cancer, which is what we're really aiming for. So why are we doing this at MGH? And I think it really comes down to this little timeline here, which is that we have incredible scientific talent. Some of you will meet Becca Larson later. She was in the Harvard graduate student program in immunology. Brilliant. She's written 20 papers in the 5 years that we've been here, and she's discovered new mechanisms that make solid tumors resistant to CAR Ts. And so we have incredible talent from the Harvard system, from the MIT system that come into our labs and generate these creative ideas that can then be leveraged for the future. We have an innovation office within our MGB system that develops and helps us maintain our intellectual property. And so we do file a lot of patents and we defend them and prosecute them. We have an internal regulatory team to keep us in line with FDA and with a lot of institutional processes to make sure that what we're doing has an appropriate risk-benefit ratio, and that we are always thinking about putting the patient first. Do they have informed consent? Does this therapy offer a good risk-benefit for the disease that they have and for the potential for treatment? We've built the infrastructure. Some of it was pre-existing from doing bone marrow transplants. This is a picture of the cellular therapy and transplantation laboratory, which is where all the cells get processed. They don't get manufactured there, but they get processed. So when the cells get collected, they have to go to a lab to get labeled and shipped to wherever they're going. And then they have to get thawed and prepped and all of that. And all of that happens in this beautiful, sterile facility. We have the patients here. And so I think that's one of our number one draws is that we have the patients, even patients with rare diseases, come here, and that really informs our science and what we're going to go after because we identify those unmet needs. And finally, I think part of the reason that we're interested in doing translation is because we think that getting these early sort of go, no go decisions will make it so that it accelerates commercialization and brings the therapies that we think are going to work directly into patients. And so we think that that offers unparalleled benefits. Now, just a little bit about IP. This is actually an older study now, about 2 years ago. We're actually a globally recognized node in this publication of a network analysis of all the patents around CAR-T. So this was 5 years after I landed here, and here's MGH, right? So you can see that the CAR-T field has evolved from just a couple of centers, and we're one of them. And so being able to generate these products and generate composition of matter IP and leverage that creativity gives us a potential downstream revenue, and also it's our science, right? So we care a lot about it, and it's our patients that we want to try to help. And this is also and I think you've experienced this just by listening to Jillian much better than I can show you with a PET scan, but this is one of our first cars that came out of our lab. So when you go tour our lab, the first CAR 37 was made like at the bench that you're going to sit at. And we tested it in test tubes. You're going to see the tissue culture rooms there. We tested it in mice. You're not going to see those. And then we did all of this process to get it to a clinical trial. And this is patient 5, who was a 35-year-old woman with classical Hodgkin's lymphoma. This is a PET scan. And so all of this white here is disease. And 21 days after getting a single infusion of this completely new CAR that we made, we created, we made, we synthesized, we manufactured, we did the whole thing, soup to nuts. And the patient had initially a partial response, and then this was an early scan at month 3, she was already in a complete response. And we have replicated this concept of a living drug, because living drugs can mean cure. When you have to give a drug every 3 weeks, you're probably not getting to cure. If the T cell, which is a live cell, can grow, like you see, it's in this blob here, right? So before the CARs, there's nothing there. After we give them 21 days later, there's a whole bunch of CAR there. That means the cells are alive and they're growing. And so this patient had done very well after CAR 37, even though she had had multiple other therapies that hadn't really worked for her. And so you can't meet this patient now, but I'm showing you this PET scan at an earlier time point. And so, and I addressed a little bit as well what some of our hurdles are and how we're trying to use our science to identify that. So we've had tremendous success in blood cancers, and there's 6 FDA approved CARs. But what we're looking for is new targets. And this is CAR 37, which is the first one that we've taken to clinical trial with the support of Ian and Izzy Loring, who are, I think, here virtually. And so we've treated 5 patients on that, and we're expanding enrollment. We've developed new CARs that target 2 different exit signs, or 2 different molecules on multiple myeloma. And so we're now getting to dose level 2 in that. We've treated 3 patients. And we're trying to address each of these problems, the 4 that I mentioned before: target, solid tumors, manufacturing, and beyond cancer, in all of these. So I've just kind of color coded the star. So we've got red for new targets. In myeloma, we're actually looking to do this multi-center studies to try to do manufacturing in multiple places. And we're also developing new targets that are double targeting, and this could have effects in lupus and other non-cancer studies. So that's going beyond cancer. And then we've developed a new platform where we have the CARs make a new drug at the tumor site. And that's going to be our approach for solid tumor. So we're trying this in glioblastoma. We're going to try it in pancreatic cancer. We have some plans to try it in gastroesophageal cancers with Dave Ryan. And we're also going to try it in a myeloid leukemia, which is AML, which is a blood cancer. We have a potential new target for breast cancer, and we're also trying to enhance the ability of these CARs to stick around in solid tumors by using CRISPR-Cas9 gene editing to really try to identify the most powerful kind of T cell that will be optimal for a solid tumor. And so we're doing a series of tests in mice now, and we're actually in the middle of doing our FDA GMP manufacturing of the tools that we will need to use in order to make the cells to treat patients with these. So you can see, if you've seen like biotech pipelines, this is probably bigger, longer, and I haven't even included all of it. And the reason we can do that is because of your help and because we're academic. So each one of these is somewhere in like the $3 to $5 million range to get to proof of concept, which is an incredible number of shots on goal that you can take in this kind of setting, where we kind of turn the crank and we have like one manufacturing process, and we can use the same kind of manufacturing process over and over again to try to get at different ways of making the CAR Ts better. So I will stop there. The phone died, so I don't know what the timing is, but I assume no one will be upset if I ended a bit early. I'm happy to take questions.
Any questions for myself?
Protect CRISPR. Are you working with companies doing CRISPR work for you? Are you able to bring that in-house?
We are able to bring in CRISPR-Cas9 in-house. Yeah, so I mean, you probably know there's like still patent fights going on around CRISPR-Cas9, but I think right now the Broad's CRISPR-Cas9 is the one that is winning. But you can use CRISPR-Cas9 gene editing in a phase 1 trial when it's research. You don't really get into like patent or like you. Down the road, you have to like have freedom to operate and you know get into licensing issues. But right now, it's about proof of concept. So we are trying to knock out specific genes, and our IP will be more around the genes than the specific CRISPR-Cas9 technology. And now there's actually multiple companies doing CRISPR-Cas9, so you could just switch. So I think we are under like a safe harbor as a researcher. In the future, I think our IP will be more around the specific CAR and the specific genes that we're editing with the CRISPR and how we're editing it, and not so much the CRISPR itself.
So thank you, Marcella. At this point, we're going to bring your guests who will take you up to Marcella's lab for a tour and some more Q&A discussions.
Thank you.
Very much.
So for our